yeah, I just touched down and now you know what's up, like, on the move, yeah, I might get a place where I can come to you, like, motivation, I might not get so much sleep tonight, rolling through the hills, looking for you still, I'm on a mission, I found her, underneath the blue skies, I found her, tangled in the hills at night, I found her, all along this endless coast, yeah, found her, underneath the palm trees where I'll be, California, found her, yeah, California, found her, yeah. I just need to get my shit in order. Open up your body and explore you. Swear that you felt this way before you. I just can't sit back and ignore this. Falling for the sunset. Yeah, I ain't had enough of you yet. I can't explain it. Californication. Hey, yeah. Underneath the blue skies, I found her, tangled in the hills at night. I found her, all along this endless coast. Yeah, I found her, underneath the palm trees where I'll be, California. Found her, California.
Hello, Year 11. Welcome to your biology revision session. So what's going to happen, Miss Sellers is going to go through spec 5 and 6, then I'll go through spec 7 and then triple content, and that will be 7 till 7:30. So over to Miss Sellers. Comments are on, but if there is any inappropriate chat, then Mr. Schuller is manning the comments and you will be blocked. Thank you.
Okay, good evening. We're going to get straight on in, okay? So just bear with us. So first thing we'll look at is homeostasis and what it actually is. So homeostasis is the regulation of internal conditions to maintain optimum conditions, okay? So it's all about maintaining optimum conditions. Now, we're going to look at some in more detail than others. So we're going to look at blood glucose concentration, how our body maintains this. So blood glucose. We're going to also body temperature. I'm hoping you all know the internal core temperature is 37 degrees Celsius. And then water levels. So these all have to be maintained. Okay, now these automatic control systems may involve your nervous responses or they can be controlled chemically. Now, your human nervous system reacts to surroundings and coordinates your behaviour. And it does this by using our, neurons basically. So we've got a neuron diagram. You need to be able to label a neuron diagram. So first of all, we've got dendrites. These are those branched endings. Your branched endings or your dendrites are there to pick up impulses. That's what they're for. Then we've got the nucleus. Then you've got your axon in the center. You've got your, are you okay? Mm-hmm. Yeah. Then we've got the axon terminal. My writing's too big, I'm sorry. And here, just around here, you have got the myelin sheath. And what this does, myelin sheath, it prevents impulses from crossing over. So stops impulses crossing over. There we go. And then this one is your cell body. Just over here. Okay. Right then, I'm going to turn over, flip it over, and we'll move on. We are going quite quick, guys, and I do appreciate that, but we've got a lot to get through. Okay, the next thing we're going to look at is the reflex arc. Okay, the reflex arc. Reflexes are automatic and very quick. They are rapid. You need to be able to write down and explain what the reflex arc is. So first of all, it starts with the stimulus. Okay, then the stimulus is picked up by receptors. Receptors just detect things. Okay, receptors just detect stuff. The impulse then travels along your sensory neuron. Across to your relay neuron. Finally to your motor neuron. To an effector, which is always a muscle or a gland. Oh, I've run out of space. Muscle or a gland. I'm going to squeeze it in there. Okay. And then finally causes a response. Okay. Now, a reflex bypasses the brain. Okay, bypasses the brain, which makes it that bit quicker. Oh, do we need to move?
We know it's shaking a bit, guys, so we're going to try and stop the shaking of the camera. Just 2 seconds.
Is it me moving there?
That's
So
what
it's
the
just
table.
one with table.
Okay.
A bit further if you can go.
Sorry, guys.
Right across.
Right across.
Yeah, right edge of table.
Yeah.
Yeah, no, it should be. It should be.
Apologies, year 11. Sorry, showing me a tissue. Grab these.
Oh no, come out. Okay. Thank you. Okay. I'll carry on. Bypasses the brain, so that makes it even faster. All right. Okay.
That's a lot better.
Good stuff. Good stuff. Now we did do the required practical, which was the ruler drop test, which tested your reaction time. So that may come up. Okay. So we did do a practical based on that. Now, I'll just have to move a little bit. There we go. Okay, the next thing we're going to look at are your synapses. I'm hoping you all know what a synapse is. A synapse is a gap between two neurons. Now, you should know that neurons do not touch each other. Okay, so the sensory neuron doesn't touch your relay neuron. The relay neuron does not touch your motor neuron. So the electrical impulse has to cross these synapses or these gaps. So if I said this one here was the end of your relay neuron, I'll just put RN. Hopefully you are aware that this one then must be your motor neuron. So what happens is, as the electrical impulse, so I'm going to put electrical there, as the electrical impulse reaches the bottom or the end of this relay neuron, it stimulates a chemical to be released. Okay, so this is a chemical. Those little orange dots are a chemical. That chemical is called a neurotransmitter. So if you're really struggling, just write chemical. Okay. That chemical diffuses, put it in capitals, diffuses across this gap, the synapse, and it binds to receptors on the next neuron. So you can see that I've drawn it. So it's binding to the next neuron. So I'll put binds. Binds to receptors. Okay, and then that stimulates the electrical impulse now to travel down this neuron. So just to reiterate and make it clear to you, it goes from an electrical impulse to a chemical impulse and then back to an electrical impulse. That's really important. Okay, so that should be synapses over with. Right, next thing then is the human endocrine system. Okay, the endocrine system is all about glands secreting hormones. Okay, and that goes directly into your bloodstream and then to your target organ. So directly into the blood and then to the target organ. All right, compared to the nervous system, the endocrine system is much slower. Oh, I keep moving it, I'm sorry. It's much slower, but it acts a lot longer. Okay, so it has a lasting effect. It lasts a lot longer compared to the nervous system, which is very quick to respond, but it doesn't last very a long time at all. Okay, so that's human endocrine system. Now, I'm just going to draw a line around this because I've started taking up a little bit more space. Here we've got the human endocrine system in my lovely diagram. Okay, and you need to be able to label them. So the first one, the one at the bottom of your brain, is the pituitary. Oh my goodness, pituitary. Forgive me, the pituitary gland. All right, that is the master gland. That's the one that controls a lot of hormones. The next one down, this one, is your thyroid. Okay, your thyroid. It looks a bit like a dicky bow, and it's in there where you put a dicky bow. Your thyroid releases thyroxine. All right, and thyroxine is there to control your metabolism and also your growth and development. Levels of thyroxine are controlled by negative feedback. All right, I'm going to change colours because we're getting a little bit cozy in here. Okay, next one is the pancreas. So this little blue one is the pancreas. We're going to look at the pancreas in some more detail in a little while. The next one down, the pink ones, are your adrenal glands. And I'm going to talk to you a little bit about them in a second. Then you've got your ovaries and your testes. Okay, so I'm just going to talk to you about these adrenal glands now. So I'll just put a little line like this. Right, your adrenal glands, they release adrenaline. All right, and they release adrenaline in times of fear or stress, and we call that a fight or flight response. Okay, fight or flight response. What happens when adrenaline is released? It increases your heart rate. This is quite important, this guy, so pay attention. So it increases your heart rate. That increases the delivery of, delivery of oxygen and glucose for respiration. Okay, for respiration. That oxygen and glucose then goes to your brain and to your muscles and allows you to either put up a fight or flee. Okay, so that's your human endocrine system. Good stuff. Right, so I'll grab my next sheets. Here we go. So I said to you about we were going to look at the pancreas in some more detail. So we're going to look at controlling blood glucose. Right, your body always likes to be what we call the normal. Okay, so if I draw this, this is our normal level of blood glucose. If you have something to eat, your blood glucose is obviously going to go up. Right, what happens is, so your body wants it to come back down to this normal. So your pancreas secretes insulin. All right, that lovely insulin. I'll underline it. Okay, what insulin actually does is it allows glucose to enter cells, to enter your cells. Okay, so that's what insulin does. So your pancreas secretes insulin. Now, your cells can only take in so much glucose. So if you have any excess glucose, then any excess glucose is stored in the liver, and it's stored as glycogen. Now, in your exam, you've got to spell these correctly, okay? So you won't get your mark, so please make note of how we're spelling them. So any excess is then stored as glycogen in your liver, and that would bring your glucose levels, your blood glucose level, back down to this normal, this nice level. All right, so then let's say you go for a jog. So we'll just write exercise or something. Your blood glucose is then going to go down. So your pancreas again kicks in. It secretes or releases glucagon. Again, you've got to spell these correctly, so please take note of how we're spelling them. Glucagon. Glucagon, what that actually does is it converts glycogen back into glycogen converts into glucose. And then that glucose goes back into your blood, okay? Which obviously increases your blood glucose back up to normal. This whole process is actually called negative feedback. So if it comes up tomorrow, make sure you mention that this process is controlled by negative feedback. Okay? All right, then. Good stuff. Okay, so people that can't control their blood glucose have diabetes, basically. There's two types you need to know about, type 1 and type 2. So type 1, it is where the pancreas fails to produce enough insulin. So not enough insulin. All right, so that's the first point you need to know. Second bit is it's characterized by you have uncontrolled, so this is how we know that you've got it, uncontrolled high blood glucose. Just put a little arrow. High blood glucose levels. Okay, and it's treated with insulin injections. Oops. Injections. It's really hard writing like this. Injections of insulin. Okay, so that's how we treat type 1 diabetes. Type 2, a lot more common. That is where your body cells no longer, they don't respond, do not respond to the insulin that your body creates. Do not respond to insulin. The way that we treat that is by a carbohydrate-controlled diet. Carb-controlled diet, and exercise regime, okay? Don't just put a healthy diet. You need to mention a carbohydrate controlled diet. And just so you know, obesity is a risk factor for type 2 diabetes. Okay. Please forgive my writing. It's terrible. Okay, and I've just drawn this sort of quick little graph over here. So you might get something like this. We've got blood glucose going up this one, time going across the bottom. Now, it might say to you, which one, person A or B, has diabetes? Hopefully, you will know that this person A has had a meal, so their blood glucose goes up a little bit, and then it comes back down to the normal. Person B, on the other hand, look, they have already is much higher, their blood glucose, but they've then had a meal, and look, it does not come back down to normal. So you would then say person B is the one with diabetes. Okay. All right. Okay, so turn over. Am I going too fast? Are we okay, or am I going too slow? I've had about 20 minutes. Oh my goodness. All right. Let's speed up, guys. We've got to go quick. Okay, next bit then, reproductive hormones. We've got estrogen, the main female, okay, hormone. And then we've got testosterone, which is the main male reproductive hormone, involved in the production of sperm. So I'll just put male. All right. Now, we're going to look at the menstrual cycle. You have to know the 4 hormones involved in the menstrual cycle and their roles. So it says we've got FSH, which is your follicle stimulating hormone. What that does is it stimulates estrogen production and it matures the egg. Okay, FSH matures the egg. LH, which is your luteinizing hormone, causes the egg to be released. That's also called ovulation. Then we've got estrogen. And that inhibits FSH or stops the production of FSH or the release of FSH and builds up the uterus lining. And then last but not least, progesterone. Okay, and that maintains the uterus lining. Then we're going to skip over to this bit, forms of contraception. Okay, they may ask you about hormonal and non-hormonal. You need to be able to tell them and give them examples of both. So we've got your oral contraceptives, your pill, for example. That is hormonal. All right, what that does is it inhibits FSH. So no eggs mature. Okay. Your injection or your implant, again, that's a hormonal example. Very similar. It stops the maturing of eggs. All right. So no eggs mature. And that one, your implant, your injection actually uses progesterone to do that. Okay. Your injection is more, and your implant is more long-term. All right, so they last a long time. Your condoms, non-hormonal. I'm just going to put NH to speed it up a little bit, all right? And this is a barrier method. Get that word in, barrier method. So it prevents the sperm from reaching the egg, okay? So prevents sperm reaching egg. Prevents sperm reaching egg. That is always a good one to write down. And your intrauterine device, what happens there is that's a hormonal, and it prevents the implant. Sorry, it prevents implantation. Prevents implantation. Okay, done. All right, next bit then, guys. is IVF. I'm hoping you're all brilliant at this. So we've got the stages down here. The mum, the female, is given FSH, and LH to mature several eggs. Okay, those eggs are then collected and fertilized by sperm. So, eggs collected and fertilized. And fertilized using the donor sperm or the father's sperm. They then develop into embryos. I'm just going to write it here. Embryos. And at the stage when they are tiny balls of cells, one or two of the embryos are inserted back into the mother's uterus. So they are inserted back into the uterus. Okay, now there's positives and negatives to IVF. I'll just get a different colour. So positives are obviously that a couple or anybody can have a chance of having a baby. The negative side to it is though that there's low success rates and multiple births. So if you implant two eggs, for example, then you're going to get multiple births, which can be a risk to the mum and to the babies. And also it's physically and emotionally stressful. So you might get asked about pros and cons to IVF. Okay, so I think I've done that sheet. I'll just change sheets. Right then. Okay, reproduction, very quickly. Two types, we've got sexual and asexual. Is that okay? Am I lined up?
A tiny bit down there.
Is that okay?
Perfect.
All right, super. All right, so sexual then is the fusion of male and female, gametes. Okay. For example, sperm and egg in animals and pollen and egg in flowering plants. In sexual reproduction then, there's mixing of genetic information. Mixing of genetic info, and that leads to variation. Whereas in asexual, there is only one parent. So therefore, there is no fusion of gametes. Genetically identical offspring are produced. And we call those clones. Okay? Call those clones. And that involves mitosis only. I'll just write that in just in case. Mitosis only. All right. So we're going to look at meiosis. All right. Meiosis is how we form gametes. So how gametes are formed. Sex cells, basically. Okay. Very quickly. All right. So the first stage is the genetic information is copied. So it makes a copy of itself, replicates. They pair up. So chromosomes then pair up. And at this stage here, DNA is swapped over. It's mixed up. Then the cell divides. So this is your first division. And then they divide again. So that's your second division. So it divides twice in mitosis. Oops, division. And you end up with 4 genetically different cells. So 4 genetically different cells. So these could be 4 egg cells or 4 sperm cells. These are called your gametes, these down here. Gametes contain half the number of chromosomes. Okay, so half the number of chromosomes. And then at fertilization, so I'll just write this, at fertilization, I'm going to spell it right, the full number is then restored. Full number of chromosomes restored. Okay, so if I just draw a quick sketch, we've got your sperm, terrible. We've got an egg, okay, fused together at fertilization. Okay, so this is at fertilization. I'll just write it there, fertilization. And then this new cell then goes on to divide. So I'll just draw lots of them, like that. And it divides by mitosis to develop that embryo. So at first, we start off by meiosis, but then when the embryo is developing, it divides by mitosis, okay, at this stage. Right. Are we okay for time?
Yep.
We are all right. Thank you. Good stuff. Okay, then. So now we're just going to go on to some key terms. All right, you've got to know these key terms. So, first of all, DNA. DNA is the genetic material and it is found in the nucleus of the cell. And it is a polymer made of 2 strands which form a double helix. I'll try it smaller right. Okay, and it's contained in structures called chromosomes. A gene, then, is a small section of DNA. Section of DNA on a chromosome. Oops, sorry, a bit wonky. Each gene codes for a particular sequence of amino acids to make a specific protein. Okay, so each gene codes for a particular sequence of amino acids to make a specific protein. Okay, so if you can ask for what a gene is, you need to write that. Genome then, your genome, it is, just one sec, I'll keep moving it, the entire genetic material of an organism. The entire genetic material of an organism. The human genome, I'll just write this here, human genome then, has been sequenced, and that is important for some reasons. I'm just going to go through them. Okay, so first of all, the genes that can be found can be linked to different types of disease. So that's one reason for sequencing the human genome, find what's linked to certain diseases. It can be used to treat inherited disorders. And last but not least, it can be used to trace human migration patterns. Oh God, what am I spelling there? Migration. Okay, so that's why the human genome is as being sequenced. Next bit then of these. Okay. All right, a gamete then. A gamete is a sex cell. For example, sperm and egg. And remember, these contain half the number of chromosomes. Okay, gamete contains half the number of chromosomes. Chromosome then is a long molecule of tightly coiled DNA. Now, in human body cells, we have 46 chromosomes or 23 pairs. That's in human body cells. Okay, just to reiterate that. Right, so what is an allele? An allele is a different version of a gene. Okay, so for example, we all have different alleles for eye colour. So it's a different version of a gene. Dominant, dominant. If an allele is dominant, it is always expressed. Always expressed. Even if only one copy is present, okay? Recessive, on the other hand, is only expressed if two copies. If two copies are present, oh dear, copies present. Oh yeah, I should just mention, we always represent these lowercase letters, okay? Dominant, we represent them uppercase, all right? Homozygous, oh dear. Homozygous, what does that word mean? Right, it's very simple. It means the same alleles are present. Homo always means, it means same. So same alleles present. So for example, I will say capital B for eye colour, let's say brown eyes. Heterozygous then. Heterozygous means different alleles present. So for example, capital B and a lowercase b, they're different, so that's heterozygous. Genotype, what does genotype mean? That is the genetic makeup. Genetic makeup of an organism. And then last but not least, phenotype, that is the physical characteristics produced. Squeeze it on, there we go. Okay, so I'm just going to super quickly do a quick genetic cross. I'll draw it down here. All right, so we're going to do a quick Punnett square for you. I always tell my classes to start off with a noughts and crosses grid, and then you cross this bit out at the top. So if we were going to take these two here, these two genotypes, we've got, let's say this is brown eyes, and brown eyes is a dominant allele. So let's put mum up here, let's say, capital B, and then we will say dad down here, capital B, lowercase b. You're going to cross these. I'm just going to straighten up. So capital B, those two. Then these ones, capital B, capital B. So, and then this one and this one, capital B, lowercase, and then this one and this one, capital and lowercase. So all four of these children, if this was brown eyes, for example, if the capital B was a brown eye allele, all of these children would have brown eyes. All right, they would have brown eyes. These would be heterozygous for brown eyes because they've got different alleles, whereas these two would be homozygous because they've got the same allele. Right. Just to I'll just say this, that most characteristics are controlled by multiple genes interacting rather than just one single gene. Okay, so there's multiple that can control your characteristics. Okay, so just I skipped something. I have, haven't I? Have I? No, I've not. I think I'm okay. I'll turn it over. So, I'll put that one there. Here we go, guys. I've got the right one here. All right. You need to know about 2 inherited disorders. The first one is polydactyly. Okay, I always spell this wrong. Like that? I think so. Something like that, yes. That means where you have extra fingers and toes. Okay? Okay? And it is caused by a dominant allele. Polydactyly is caused by a dominant allele. The other one you have to know about is cystic fibrosis. Okay? And this is where it affects basically your cell membranes. Causes lots of mucus to build up in your lungs and things like that. And that is caused by a recessive allele. Okay, now we can actually carry out what's called embryo screening, which is here. You have to know the positives and negatives to embryo screening. So, positives are, I'll just move it so it's all nice and neat. You can find out if healthy embryos are going to suffer from a certain disease. So, healthy embryos are going to suffer, are going to carry that disease, suffer from the disease. So that's a positive to doing screening. You can stop faulty genes from being passed on. So that could be seen as a positive. Faulty genes not passed on. And it would improve your chances of having a healthy child. Improve chances of having a healthy child. But there are some downsides. There are some negatives to embryo screening. First one is there's ethical issues. Okay? You are going to be killing basically a faulty embryo. And is that correct? Some people say not, some people say it's okay. It's an expensive process to carry out embryo screening. Oh God, what am I writing here? Expensive. It's very expensive. And then obviously you have an operation for this, which could lead to infection. So that's the negative side to it. All right. Now we can also, well, the next thing, I should say, is that we're looking at sex determination. Right, the human body cell. Remember what I said, 23 pairs of chromosomes. All right. 22 of those are control your characteristics and only 1 control your sex. Females have XX chromosomes, whereas males have XY chromosomes. You've got to know that. Okay, so if I were just going to draw you a little Punnett square again, draw your noughts and crosses grid, cross that one out. Put your females at the top, XX, put your males at the side, XY, and then look what we do. Cross them over. So XX, XX, XY, XY. Okay, now you can see, look, 50%, we've got 50-50 chance of either being a female, which are the top 2, and a male being the bottom 2. These are this is the only time when your Punnett square will have different letters. So an X and a Y. Your other Punnett squares will all have the same letter, whether it be uppercase or lowercase. Okay, so if you've got different letters in there and it's not talking about sex, it isn't right. All right. So next thing then, variation. So variation is just differences in characteristics. Okay, so differences in characteristics. That's what variation means. Miss, are we okay for time?
I'm pushing it. Just
Oh
no. It's all right.
No,
We'll
it's
stay
fine.
late.
no. Are we sure? Are we sure?
Yeah, we're fine.
Okay, it can be caused by inherited genes. Okay, for example, eye colour can be controlled by the environment. So, for example, scars, or a combination of both genes and the environment. So, for example, weight, your weight. All right. Now, variants arise from, so all variation arises from random mutations. Random mutations. Okay, and we can increase those mutations by going out in the sun too much, so UV rays, and also chemical carcinogens. Okay, so mutations can be increased by chemical carcinogens. Right. Turn it over. Lovely diagram. We're getting there. We're getting there now. All right, I'll speed up super quick. Right, evolution then is a change in inherited characteristics. Change in inherited characteristics. In a population, I spelled characteristics. Okay, in a population over time. So in a population, abbreviate stuff, over time. Through natural selection. Okay, this can result in new species forming. So evolution can lead to new species forming. You have to know what a species is, and a species is where two organisms can reproduce to give fertile offspring. Can reproduce to give fertile offspring. That's what a new species is. Okay, so the theory of evolution states that all species evolved from simple life forms that developed over 3 billion years ago. All right, now we're going to look a little bit more closely at what natural selection is. So very, very quickly, very briefly. Step one, there's variation in a population. In a population. Remember that variation is down to random mutations. Step two, there's competition taking place between these organisms. The third bit, the phenotypes that are best adapted go on to survive. We call that survival of the fittest. All right, I'm not going to write it, but it's survival of the fittest. And therefore, those organisms that survive go on to reproduce and pass on their gene. Pass on their genes. And that is called natural selection. Now, the evidence for evolution, we have evidence for evolution, and the evidence is fossils, okay? And also looking at antibiotic resistant bacteria. By studying those, we can see that evolution is taking place. Okay, so that's your evidence. I'll just write that there so you know what I'm on about. Okay, right, selective breeding then. I'm so glad that I printed this off. Selective breeding is where humans breed plants and animals for particular genetic characteristics. Okay, so for example, food crops or domesticated animals. So in this example, we've got a herd here. And in this particular case, we want to breed for the biggest cow. So we've got this herd. We're going to take the two biggest cows, in this case, this one and this one, the bull and the cow. They have offspring. Now, from those offspring, we then take the biggest of them. So we take out the biggest two and we actually breed from there. And then we repeat that process. Okay? So it continues for generations until all the offspring have that characteristic that you want. Okay, so that massive, that large cow. But obviously, this leads to inbreeding. Okay, which is not a good thing. But what is good about it is we can use selective breeding for crops to make them disease resistant, which is obviously great. We can make it so we can have more meat and more milk produced. We might want more like a gentle nature. So you might want dogs that are more gentle. Or you might want to have like large fruit or large flowers or something like that, large flowers and unusual flowers produced. So we can use selective breeding for all those things there. Okay, all right, I'll just... All right, now we're looking at genetic engineering. Genetic engineering is where you introduce a gene from another organism, to give a desired characteristic. To give a desired characteristic. Okay. There we go. So, for example, in plants, we can genetically engineer them and make them resistant to insect attacks or resistant to disease, give them bigger fruits. And we can also genetically engineer bacteria to produce insulin for diabetes. Okay, now this is what we've got here. We've got the stages of genetic engineering. So the first stage is that we remove, I'm just going to sort of skip down to this bit. We isolate and remove the gene responsible for insulin production. That's the first thing that we do. So we take out of a human body cell, the gene responsible for insulin production. We then take a bacteria and we remove a plasmid. That is a plasmid. And a plasmid is a vector. Okay, just a carrier. And we cut open the plasmid using what we call a restriction enzyme. Okay, a bit like a pair of scissors. All right, so we've cut it open. And then obviously what goes off here is we insert the insulin gene into the plasmid. And to do that, we use a ligase enzyme, using ligase. Okay, using a ligase enzyme. I'll write that enzyme in. Okay, then that plasmid is inserted back into the bacterium, and we all know that bacteria are fantastic at reproducing. Reproduces, and therefore produces the insulin, and that is what we want. Okay, positives and negatives to genetic engineering then. Positives, we get increased yield. Okay, so more crops. If they're resistant to disease, they're going to get more crops, aren't we? Okay, therefore farmers get more money. You can make plants resist insect attack. Insects, increase insect. Oh God, increase insects. What should I write? Attack. I'll put resist. Wrong thing. Resist insect attack. Sorry, what did I say that one? And then obviously we can produce increased supplies of insulin. So they're all good things, but there are negatives to it. We're not sure of the effect on a population of wildflowers and insects. Okay, so we don't know how our genetically engineered crops can affect any wildflowers or any insects. And what about the health impacts on us? What if we eat these genetically altered crops? What's the health impacts on us? Okay, we've not explored it enough yet. We're getting there. Okay, very quickly then. Fossils. What is a fossil? A fossil is the remains of organisms from millions of years ago. From millions years ago. And they're found in rocks. Okay. Right, they can be formed. So fossils can be formed when? Conditions are not correct. So incorrect conditions. So if it's too cold, there's not enough oxygen. Incorrect conditions. For example, temp 2. Another way is parts of the organism are replaced by minerals. Replaced by minerals. Another one is preserve traces of organisms. For example, footprints. Traces, e.g. footprints. And the last one is hard parts take a long time. Hard body parts, for example, teeth, bones, take a long time to decay. So, teeth and bones. Why is the fossil record incomplete? Well, it's because the early life forms are soft-bodied, or were soft-bodied, I should say. They're soft-bodied, and then also due to geological activity, a lot of that evidence has been lost. So if they're soft-bodied, then they decay way too quick to form a fossil. So that's why the fossil record is incomplete for lots of species. Extinction then, very easy. That means no remaining individuals. Get that word in, individuals. Other species remain. Okay? reasons that organisms can go extinct. Think about it. What about disease? Loss of habitat? Hunting? Competition? New predators? Something like that. So these are all reasons why organisms can become extinct. Just draw a line down. Okay, very, very quickly. Oh, I've just not said that. Okay. Sorry.
It's okay.
Thank you. Okay, resistant bacteria then. Resistant bacteria evolve rapidly because they reproduce at such a fast rate. So talk about reproducing. Got a bit of a diagram here. So we've got a colony or a population of bacteria, and you can see that a random mutation has taken place. So it's totally random. And that random mutation makes that bacteria antibiotic resistant. Okay. Start taking some antibiotics, and look, you can see that all of the bacteria are killed apart from this antibiotic resistant bacteria. This one survived, and therefore that one goes on to reproduce. Write it up here. Reproduce. And it basically spreads the antibiotic resistant gene, and there's no effective treatment. So, and no effective treatment to even get it back down. No effective treatment. Okay, for example, an example is MRSA. That is now an antibiotic resistant bacteria. Now, things are in place to try to reduce this. Doctors cannot prescribe for non-serious illnesses or viruses. So to try to reduce it, doctors can't prescribe. Not prescribe. Viruses are non-serious illnesses. And also you have to complete your course of antibiotics. Okay, new antibiotics though take a really long time to actually to develop, they're very, very costly, and they cannot keep up with the new strains that are actually developing. Okay, we're on to his last thing. We're on to it. All right, last bit, classification then. So traditionally, organisms were classified into groups based on their structure and characteristics, and this method was proposed by Carl Linnaeus. Okay, so that was based on structure and characteristics. Okay, he came up with this. All organisms are classified using this system: the kingdom, the phylum, the class, the order, family, genus, species. Easy way to remember it: king, prawn, curry, or fat, greasy, sausages. Okay, easy way to remember it. Now, I've just put an example here of a binomial name. So this is an example of a binomial name of a species or an organism. Now, the first part here, the first part of the binomial name is always the genus. Okay? So if you were asked that, you'd write Spodoptera here. That's the genus. The last part of the binomial name is always the species. And I'm hoping it'll tell you something and you'll be able to decide what kingdom it belongs to. Does it belong to a plant kingdom or does it belong to the animal kingdom? Okay, so hopefully First part is always the genus, second part is always the species. Now, due to improvements in microscopes, and now we have evidence of internal structures and chemical analysis, we've got a new model of classification. And that new model was proposed by another Carl, and he's called Carl Woese. The new model classifies organisms, I should say, into what we call archaea, and these are primitive bacteria. And they normally live in extreme environments. Oh God, what am I writing there? Extreme environments. Bacteria, that's your true bacteria. And then your eukaryota, which are your protists, your fungi, your plants, and your animals. And sometimes they may represent this using an evolutionary tree. Okay, so I think I have covered now spec 5 and spec 6. I don't know if there's anything else. Are we okay? All right. So we're going to go for a 5-minute break then, guys. Get yourself a drink. Thank you, miss.
I want to play some tunes. Are you ready?
This is going to be really embarrassing.
Well, we can't play music because we're not connected to the cabling, so I thought I could play some accordion music.
Mr. Shill's actually putting an accordion
on.
on.
You know, I learned to play Twinkle Twinkle Little Star on this,
Oh, God.
but now I can't remember.
Thank you for my drink.
That's okay. I can't remember. I can't how to play it, but I will attempt some.
No more, please. That is really enough.
I'll get my backpacks out for the next
It's
break.
enough, thanks. Thank you very much, Mr. Schuller.
Any time I'm available for bookings.
Right, year 11. Over to me to look at spec 7. I'm sorry I sound like I've got a cold. It's just my hay fever. I know you're all concerned about me. I've seen some comments about spec 7 being the hardest one. It's not the hardest one. There are just more definitions to learn. Also, this is a spec that you've literally just done. So what we're going to do, we'll move straight into spec 7. And the most important bits in here are your definitions. So you need to know, oh, it's hard to write like this. You need to know what an ecosystem is. An ecosystem is the interaction of biotic and abiotic factors. And you need to know what biotic and abiotic factors are. So if we have a look at biotic factors first. So biotic factors are living things that can affect distribution. And abiotic factors are non-living things that can affect distribution, but I'm not writing it. Just can't bother. So if we want to classify biotic and abiotic factors, which you could be asked to do, we'll draw a quick table. This would be a nice question if you got it. We'd have biotic on one side, abiotic on the other. So examples of biotic factors You've got things like food availability. You've got things like predation, things like competition. Abiotic factors, so non-living factors, you've got plenty of these. So you've got things like light intensity, temperature, moisture levels, soil pH, wind intensity, oxygen levels. So there's lots and lots of different factors that could affect the distribution of organisms. You could be asked to classify these factors into biotic or abiotic parts. So if we have a look at the next part of this, we need to have a look at the competition that could occur. So both plants and animals compete with each other for certain things. So put a thread in competition. And if we have a look at the things that plants and animals might compete for, so plants, obviously plants could compete for light, they could compete for water, they could compete for space to grow, and they could compete for minerals from the soil. Be very careful when you're using the word minerals. Sometimes it will let you use nutrients, but I would never use it. So use the term minerals from the soil. Animals, if you have a look at animals, animals could compete for a mate, they could compete for food, could compete for territory. If I can spell it. So they're the things that animals and plants could compete for. As well as competition, you need to be aware of what the term interdependence means. Interdependence is how living things, I should say organisms really, living organisms depend on each other for things like food, seed dispersal, pollination, and things like that. In a stable community, so in a stable community, these factors are all kept in balance. So when one of the species may die, another species in that ecosystem could replace them. So a stable community, all the things, abiotic, biotic factors are in balance. So that's the first part of spec 7. So if you have a look at the next part's the nice bit in my opinion. So it's learning about adaptation. So if we just move those out of the way, got a nice picture of a gazelle. Could have been anything, I've chosen a gazelle. Adaptations is one of the easiest parts. You just need to be able to recognize how an organism is adapted to the environment in which they live and the features that allow them to live there. For example, nice easy one. The gazelle has antlers. That's a description. The explanation is for defense. So that's antlers for defense. If we have a look at this one, they've got long legs. Should be pretty clear from a picture of an animal that this is a prey animal. They need long legs to escape predators. Another example that you could have, if we look here, they've got large ears. The explanation is to listen for predators. There are lots and lots of different things. If we have a look at the ones that I've drawn on there, they're all adaptations for prey animals. You could be asked for adaptations to hot and cold. I've not picked a very good animal to look at for adaptations for hot and cold, but I'll put a few on for you. So it's got long, thin limbs. This will increase the surface area to volume ratio, which will increase heat loss. Mr. Catchall, this is just for you. Just had a little text message in. Okay. It's also, if we have a look here, try not to write in that part, it will have thin fur. This is to reduce the insulating effect. So the ones that I'm just going to put a little star next to, so these ones are adaptations to the climate. The other ones are adaptations for defence. As well as general adaptations, you could be asked about any animal, any plant. It is just a bit of common sense. Make sure you make a description and an explanation. You could be asked, and Miss Sellers touched on this briefly, about extremophiles. These are, firstly, archaea that live in, clues in the name, extreme environments. So I'm not writing very straight. It's hard to write on here because we're trying to make it not wobble for you. The examples of the I spelled that wrong, put another name on it. The examples of extreme environments, you've got hot springs, you've got salt lakes, you've got deep sea vents. These are the places that you will find extremophiles. How they can live in those areas, they have got functional adaptations. For example, one of these functional adaptations that their enzymes can withstand high temperatures. Excuse me. So there are functional adaptations. There are also behavioural and structural adaptations. So if we just look back at the gazelle, a behavioural adaptation would be that it lives in a herd. So that is behavioural. A behavioural adaptation is to do with the behaviour of an organism. So the gazelle would live in a herd to reduce the chance of it being eaten. If we look at structural adaptation, structural is basically about the body, about any features that it has. So the antlers would be structural. You could be asked to define those. Okay, so that's a little bit about adaptations. We'll next have a look at levels of organisation. So we'll skim through this quite quickly because it's nice and easy. So there are things called feeding relationships. And to illustrate this best, I'm going to write a little food chain. So if we have a look, we'll start with grass. Then we'll have rabbit. And then we'll have fox. Very simple food chain. If you ever have to draw a food chain, it doesn't mean draw all the tiny little animals. It just means you write them down. You need to be able to identify the levels in this. So grass, this is a producer because it uses light energy to create carbon compounds. The carbon compounds are then eaten by the rabbit. This is the first consumer, so we call it the primary consumer. The rabbit is then eaten by the fox, which will be, in this case, the secondary consumer. Okay, you can sometimes in certain food chains have a tertiary consumer, but it's unlikely in a lot of cases because the energy available gets less at each stage. Your producers are usually, and I state that clearly, usually green plants or algae. Not always the case. So your producers are green plants or algae. These are eaten by a primary consumer. These are then eaten by a secondary consumer. Okay, You can also be asked about how do we actually sample for this. So this links into the required practical, so I will go over it. So you could be asked for your ecology RP. You could be asked how to use random sampling and also transect sampling. Okay. I'll do random sampling up here. So if we look at random sampling up here, this is used if you want to know the abundance of a certain species on a big area. So imagine we've got a school field and the dots are daisies. Would you count every single daisy? No, you wouldn't. It would be too time consuming. So what you tend to use is a quadrat. So in this investigation, you would randomly sample. So you would throw a quadrat at least, I'll say 3, should really be 5, at least 3 times. You would count the daisies in each quadrat, and you would calculate the mean. What you need to remember when you do this is you need to do this stage, so here it needs to be totally random. This avoids bias. Once you've calculated your mean, you need to multiply your mean by the number of quadrats that would fit in the field. In the field. And to know how many of it would fit in the field, you obviously need to know, oh gosh, that's a wonky line, the area of the field, you know how to work that out, base times height. You also need to know the area of 1 quadrat. When you know those two areas, you just divide the area of the field by the area of 1 quadrat, and it will give you how many would fit into that field. And that is random sampling. You can also be asked about transect sampling, and this is the one where we took you out and it was a bit of a farce because we couldn't find anywhere to do it. So if you imagine you've got a tree line or the edge of a forest, and we want to know how the distribution of an organism changes as you move away, you don't throw anything randomly. What you do, you set up what we call a transect, and then you place a quadrat at set intervals and you estimate the percentage cover. So I would place a quadrat there, right at the edge, there, every meter, and I would estimate the percentage cover. Okay, that's a required practical that we would have done outside. When you're looking at this, you need to compare it against an abiotic factor. So we looked at the light intensity. So the light intensity as you move away should get brighter. So there'll be less shade. If there's more light, there's going to be more photosynthesis. Can't fit it all on. So you're going to make more glucose. So this means that will be more protein to grow. That is the explanation. So that is your required practical that you could be asked about. The next little bit, guys, you need to know about cycling of nutrients, well, not nutrients, cycling of materials. I'll just follow that so it sits down a bit straighter. So if we have a look first at the water cycle. You've covered this in chemistry, so I'm not going to spend ages on it. So the water cycle, pick a nice pen. When the sun shines down on the water will evaporate. Can't even say that word, can't spell it. Best I can do. Evaporate, is there a U in it? Never mind. So the water evaporates. When it reaches the colder air at the top, the water vapor will condense. It will fall as rain, or we can use a proper term, precipitation, and then it will collect. So these are the main 4 stages of the water cycle. The sun shines onto the water vapor evaporates, it then condenses, falls as rain or snow or sleet, and then it collects again. The water cycle is important because it provides fresh water for plants and animals. That's the basics of the water cycle. The next one, the one that I like to try and draw for you all, is the carbon cycle. So Horizon students, you've probably seen my atrocious drawings of this before. We're going to try it again. So what we've got in the atmosphere, we've got, we'll call it a bank of CO2 gas.
Quick question, Miss Nurse. Do we need to know about mRNA?
Which, is it set 1, separate?
Not sure.
It does say that you don't need to know the detailed structure of it, you need to be aware of it, but that'll be when we cover the separates only content.
Lovely, thank you.
Okay, so the CO2 in the atmosphere is taken in by green plants and algae in a process called photosynthesis. Okay, because plants are living things at the same time, I'll do this in a different arrow, they are also respiring. So they take in CO2 in photosynthesis, but then they release it in respiration. I'll just put resp. Okay. Along will come a small animal. I'll attempt to draw a little rabbit of some sort. And the rabbit will eat the carbon compounds in the plant. So the rabbit eats carbon compounds that are in the plant. And these carbon compounds, you've got glucose, you've got protein, you've got starch. Any of these, sorry, I know the paper's shaking a little bit. What will happen then, that's your primary consumer. Along will come something a bit bigger. Gosh, disgusting. Try and draw a fox. So a fox will come along and it'll eat the carbon compounds in the rabbit. This is passing the carbon along. Obviously, whoa, chicken pens. Obviously, the fox and rabbit are living things, so they are releasing CO2 when they respire. We've also got another aspect of this. The fox will go to the toilet. I've no idea why I've drawn it purple.
Some beetroot, maybe.
Some beetroot, too much beetroot. And the feces and urine will decay. This is also the case if any of these actually die. So we'll have death here. So many of these things actually die. They are all, or the remains are broken down by bacteria. So remains broken down by bacteria. Bacteria are obviously living things, so like with anything else that is living, they are releasing CO2 back into the atmosphere in respiration. Last little bit on this is more from chemistry. As well as all this natural or these natural processes going along, you've also got factories burning fossil fuels, so combustion releases CO2 back into the atmosphere. Okay, so back to your cycling or how materials are cycled. We're going to move on now and have a look at the next part of Spec 7. Sorry, we'll try and speed it up because I know it's getting a little bit late. The next part is about biodiversity and our interaction really with the environment. So, biodiversity. This is the variety, that's a key word, variety of living species or organism species, I don't want to tell you the wrong word, variety of living species on Earth or in an ecosystem. So that's the definition of what biodiversity is. Obviously, a high biodiversity high biodiversity is good. That's what we are aiming for. Main reasons for it: species would be useful to us. For example, we could get medicines from them, and also we have got what we call a moral obligation to preserve species for future generations. Okay, so the overall impact of humans on the environment, on biodiversity, is massively negative. We tend to go into places and destroy everything. So humans negatively influence biodiversity. We're going to look at the ways in which we do this and how we're trying to stop it. So the first little bit of this is about waste. The amount of waste on Earth is increasing for two reasons. There's an increased human population and also an increased standard of living. And that's the reason why there's more waste being generated, and we need to also work out how we're going to store this. With more waste being generated, it increases pollution. So there's also more pollution. You need to be aware of the areas that can be polluted. I'm not going to go over this in masses of detail because I know you do it in chemistry as well. So the areas that can be polluted, you can get water pollution, you can get air pollution, and you can also get, sorry this is tiny, land pollution. The main way in which water is polluted is from sewage and fertilizer. The main ways that air can be polluted is by smoke or particulates, and also by acidic gases. Acidic gases, things like sulfur dioxide, which are generated from burning fossil fuels, can lead to acid rain. Also, the main ones on land is obviously landfill, which is not nice because you can get runoff of water, which can then pollute waterways as well. So a bit of a double whammy. Please be aware that pollution kills plants and animals. If plants and animals are being killed, this will obviously decrease biodiversity. That's a key term. Always get into your answers from Spec 7. What is going to happen to biodiversity? Excuse me. The next little bit that we've had some questions on really is about land use. So you need to be aware of the ways that humans use land, and there are four main ways. So building, farming, quarrying, and our favourite that we like to do, dumping waste. Please be aware that you could be asked why. Why are we using more land? And that will always come back to this, because of an increased human population and standard of living. Linked to this, you need to know about peat. Peat is formed from non-decayed plant material. It is non-renewable because it takes millions and millions of years to produce more peat. However, people use it as compost because it holds water very well. We also use it as fuel. You might think, well, if it's a good fuel and it makes a good compost, we should be using it. Negatives: it decreases biodiversity, as peat bogs are habitats to lots of species. And if you burn it, it obviously releases CO2, which contributes to global warming. Okay, so peat, it is a good compost, it's a good fuel, but unfortunately, if we are digging it up constantly, burning it and things like that, it's releasing CO2, which contributes to global warming. And as well, as soon as you start digging a bit of peat up, you expose the rest of the peat to oxygen. This means that it will start to decay. As anything decays, as we've learned from a carbon cycle, it releases CO2. Okay, last little bit, guys. We need to have a look at deforestation and global warming. So, deforestation, hopefully you know what this means. Deforestation, it is the large-scale destruction of forests. I think, well, why are we doing it? Obviously, we know we need plants, we need trees to take in CO2 to maintain the carbon cycle. We need to know the reasons why. Large-scale deforestation mainly takes place in tropical areas, and it is a massively negative thing to do. The reasons for it, and you need to know these, provide land for cattle farming. Don't know if I put a cross there, never mind. So it provides land for cattle farming, land for rice fields, land for growing crops, for biofuels. These are the reasons why. You need to know the link between deforestation and global warming. So I'll mention global warming here. If we are cutting down trees, there is going to be less trees to take in CO2. And as you know from chemistry, CO2 is a greenhouse gas. If we are farming cattle, lots of cattle release lots of methane during digestion. As you know, methane is a greenhouse gas. Rice fields also release, I know my writing's going all over, sorry, methane, which is a greenhouse gas. And finally, if you are growing crops for biofuels, when you burn biofuels, you release CO2. So deforestation has it's like a double whammy. You're taking the trees away, which means that less CO2 is going to be absorbed by the trees during photosynthesis. And also, any of the reasons why we cut trees down release more greenhouse gases into the atmosphere. I'm not going to go through the process of global warming because you should know this from chemistry. If you don't, then get your revision guide out. So global warming, you need to know some of the biological consequences of it. So the main biological consequences of global warming are linked to what it does to temperature. So global warming, as the name suggests, will increase temperatures on Earth. This could have two main ways of affecting everything. So it could melt ice caps, which could increase flooding. If we've increased flooding, we've decreased the habitats that are available, which would decrease biodiversity. On the other hand, if we've got increased temperatures, you could also get more droughts. I forgot to spell droughts. I hope that's right. Sorry if not. That looks like drafts. How do you spell drought? That's not drought. Is that all right? We know what I mean. There's going to be less water.
D-R-O-U-G-H in it?
Yeah,
Yeah.
it's then drafts as D-R-A-U.
Yeah. O-U-G-H.
O-U.
Yeah.
Right, we changed
No,
it.
they're not writing it.
Yeah, you get multiple comments here.
Guys, it's Sunday night. I'm dying of hay fever. I think it should be a bit more
Not helping you out, not helping you out.
All right, I'll
Give you
take
some
it back.
assistance.
Helping. Span corrections.
I'm going to say, there's no spag assessed in this exam,
Right.
you're okay.
So there's decreased water availability, which will obviously mean less photosynthesis, as that is needed for photosynthesis. So this could mean there will be less food available for animals. Linked to this, and this is one of the things you need to know for your exams, if temperatures increase, it makes birds go a little bit weird. So birds could lose the ability. Are you interested in this show? Why are you laughing at your copy? Your text messages. Lose the ability to migrate because they will not know which time to migrate. So you'll get birds freezing to death because they'll have not migrated in time. So birds will not be able to migrate. It's very, very sad. Last little bit, because it's not all doom and gloom. Humans are trying to maintain biodiversity, so we have positive and negative interactions on the environment. So what I want to go through is just some of the positive things that humans are trying to do. We've got breeding programs. Remember that video of the panda that we showed during lesson? There are breeding programs to help breed more endangered species. We're also trying to protect rare habitats, such as peat bogs, so there are laws in place. We're also trying to put in field margins. This means that instead of just having massive open fields, we've got little hedges that animals can live in. We're also trying to recycle and reduce deforestation. That's some of the positive stuff we can do. And mine looks so messy compared to Miss Sellers. She's very artistic. That is everything in Spec 7 for Trilogy students. The rest of it, and there is a lot, is Set 1 separate. So has anyone got any questions before we have a 2-minute break? No more accordion, I promise. Before we move on, any questions?
And at the moment, I'll keep an eye out. Can I get the bagpipes out?
No, but nothing.
I play guitar, which I actually can play. I genuinely can.
Oh,
But I haven't got a string. I snapped a string. So I need to restring it.
Okay. Right, Year 11, I hope that was useful for you. Once we finish the stream, we will be uploading it, and we haven't used any copyrighted material this time, so...
We have got a question.
Go on.
What is the definition of tropism?
That's Set 1. You'll have to wait until I go through it when I talk to Set 1 students. Thanks. Know your specification. I shall not know. Right, 2-minute break. Biology separates is coming up. So if you are a Trilogy student, you need to turn off now because the rest of it is just going to blow your minds. So Set 1, get ready. Okay. Okay, right, year 11 separates. I have got so many pieces of card to go through, but I don't think we're going to be able to get through everything. So I'll go through the main details and then you can ask me if you want me to go over anything else. Okay, so if we just look at the Spec 5 separate content first. So the brain, the areas of the brain that you need to be able to highlight, we have got here cerebral cortex. The cerebral cortex is involved in memory. So if you damage your cerebral cortex, you are going to have difficulty with memory functions. Okay? The next part that you need to be able to label is the cerebellum. Cerebellum is involved in movement. So if you damage your cerebellum, you might have difficulty in moving. Okay, so you could be asked what brain damage has this person got if they can't remember something, and you would need to say, right, they'll have damage to their cerebral cortex. Okay. Last little bit, what we've also got is the medulla, and this is involved in your basic functioning, such as controlling homeostasis. You can also see on this diagram, you've got the pituitary gland. Just label it there, okay? I've put in a little diagram of the eye. I'm not going to write it all on you. I've got this in your revision, guys. I'm just going to point to the main parts because it'll just slow it down if I write everything on. Obviously, you've got the lens here. Your lens can change shape to alter the refraction of light. At the back of the eye, all of this part here, that's your retina. This is where the image forms. You've got the optic nerve. So the optic nerve receives the image and sends the image to be processed to your central nervous system. What we've got covering this here, you've got your cornea. The whole here, you've got the pupil. You've got your iris, which controls the size of your pupil. As well as this, you have got your suspensory ligaments and your ciliary muscles. You've also got circular and radial muscles, but we'll have a look at this next when I show the next part of the eye. Okay, so if we have a look at this one I prepared earlier, sorry, it's got a bit wonky. You need to know how the eye responds to bright and dim light. If you are in dim light, your pupil will enlarge. This allows more light into the eye. If you are in bright light, your pupil will constrict, which lets less light into the eye because you do not want to damage the retina. And how this works, it's involving different muscles. So you have got here the ones around the outside that are going in a radial pattern. You've got your radial muscles. And here on the inside, like a circle, because they are a circle shape, you've got circular muscles. These are antagonistic pairs, which means they work in opposites of each other. So if you move into dim light, what happens is your radial contract. If your radial are contracting, it means that your circular must be relaxing. If we look at the second part of this, so if you go into constrict, bright light, so if you move into bright light, what happens is your circular contract. If your circular are contracting, your radial must be relaxing, okay? Because they are antagonistic. The next part of this about the eye is called accommodation. Accommodation in the eye. So what needs to happen? This is how you focus on close and distant objects. So if we have a look at the object that is distant first. So if we want to focus on a distant object, what needs to happen? Your ciliary muscles. This pen's a bit thick. No, I don't. I'm getting myself confused. Your ciliary muscles relax. Your ciliary muscles work antagonistically with your suspensory ligaments. So if your ciliary muscles relax, your suspensory ligaments pull tight. Sorry, I know it's shaking, I'll move it in a minute. So your suspensory ligaments pull tight, which makes the lens thinner. This means that it only slightly refracts the light, so the light is not refracted as much, and this makes sure that it focuses on the retina. So this is for a distant, hold my paper down, object. If we are looking at an object that is close, I'll do it in a different colour. Sorry, I've got my hand on it. I'm just trying to hold the paper down a bit because I know it's waving about. If we look at a close object, your ciliary muscles contract because your ligaments work antagonistically. This means that your suspensory ligaments loosen. If your suspensory ligaments have loosened, the lens becomes thicker, which means that it refracts the light more strongly. So this means that it will again focus on your retina, which is at the back of the eye, which is where you form your image. I am conscious of time, so I'm just going to very quickly look at the two common defects in the eye. So what we've got long and short sightedness, but you need to use the correct terms. So we've got myopia and hyperopia. In myopia, what happens is the image focuses before it hits the retina. In hyperopia, the image would focus after it hits the retina. So what you need to know how to correct these two vision problems. So if we have a look, to correct myopia, you need to use a concave lens. And to correct hyperopia, you need to use a convex lens. What these do, they make sure that the light actually focuses at the retina at the back, of the eye. Okay, that's the information about the brain and the eye. Next little bit, I'll try and get through this, so it's a bit wonky. You need to know about how body temperature is controlled. So you need to know about thermoregulation. So your body temperature is controlled by the thermoregulatory center in the brain, and how this regulates your temperature or monitors your temperature. It's got receptors for blood temperature. So as the blood flows through, it monitors the temperature of it, and also it receives information from the skin. So it receives info from the skin. That's your thermoregulatory center in your brain. Okay. What you need to know in a lot of detail is what happens when you are too hot and when you are too cold. So I'll just try and make this faster a bit. So when you are too hot, if we have a look at this, you start to sweat. So sweat is produced. When sweat evaporates, it uses thermal energy to change state. So this takes away thermal energy from the body. As well, what happens? Your arterioles or blood vessels leading to the skin dilate. Get it out of your head so that anything does any moving about, because it doesn't. And if you write that in your exam, you will not get any marks at all. The arterioles dilate. This lets more blood through to the capillaries close to the skin edge. So you radiate more heat. So nothing is moving about. The arterioles dilate. This lets more blood flow through to the capillaries near the surface of your skin. So you radiate more heat. When you are cold, obviously you do not sweat, your arterioles constrict, and this reduces the blood flow in the capillaries. Nothing is moving closer or away from the skin. You are just reducing the blood flow, so there will be less radiation of heat. In addition to this, everyone, you also need to know why you shiver when you are cold. So if you shiver, well, if you are cold, you will start to shiver. Is it one or two of these? Never mind. Shivering is caused by muscle contraction. In order to let your muscles contract, you need energy. This energy comes from respiration. A lot of the energy that is released in respiration is released as thermal energy. So when you shiver, your muscles are contracting, energy is being released through respiration, and a lot of this energy is thermal energy, which will help you to warm up. Okay, so that is how to monitor and control your body temperature.
Couple of questions, Miss Nick,
Yeah.
just so I've got a bit of a break there. First one, do you have to say arterioles?
No, you can say blood vessels leading to the surface of the skin.
Next question is about just asking for about clarification on, is it vasodilation and vasoconstriction?
Yeah, you can use those terms in your answers. They are included in your spec. So if you want me to add it on, dilation. So it's called vasodilation, and this side is vasoconstriction. All right. Be very careful as well to remember you can never say that these capillaries constrict or dilate. They're only really tiny, so you need to refer to the blood vessels or arterioles leading to the surface of the skin. Okay. Right. Excuse me. Next little bit on this, we have to look at the kidney. There's been a lot of asking about this, so we'll just go through it very, very quickly. Your kidneys are involved, obviously, in the production of urine. So what happens is blood enters the kidney. The first stage is filtration. In filtration, what happens is small molecules, e.g. glucose, amino acids, water, ions, pass through. Large molecules such as proteins, blood cells stay in the blood. Okay, so they don't pass into the kidney. So stage 1 is filtration. Stage 2 is reabsorption. You need to use very specific terminology here. So it is all the glucose that is reabsorbed, and it is some ions, water, basically as much as is needed. So your blood is filtered, small molecules pass into your kidney, and then you reabsorb all of the glucose back into the blood, because you do not want to lose glucose in your urine. Stage 3, which is down here, is simply urine production. So what remains in your kidney there that is filtered off, it is urea, water, and some ions. Okay, so that's what will go to your bladder as urine. That is the kidney. So the most important bit here, and where you might lose marks if you don't say it, it is all the glucose that is reabsorbed. What I just want to look at with you quickly, the reabsorption of water is controlled by negative feedback, by a hormone called ADH. So with negative feedback, the hormone ADH controls how permeable your kidney tubules are to water. So we aim to keep your water content normal. If you drink too much water, your brain will notice that the water content of your blood is too high, it will produce less ADH from the pituitary gland, so your kidneys will reabsorb less water into the blood, so more will be lost in urine. If we go the other way, if you've eaten too much salt or you've sweated too much, your brain will notice that the water content of the blood is too low. It will make more ADH. Your kidneys become, or your tubules become more permeable, so you reabsorb more of the water. This means that you make less but more concentrated urine. And this is an example of negative feedback. Okay. Link to this, I know some people were asking about this as well. In your spec, you need to know, just hold it over so it stays flat or tries to. You need to know the basics of dialysis. So obviously some people have problems with their kidneys. The main way for it to be treated is using, well, one of the ways is a kidney transplant. Unfortunately, there aren't always enough kidneys available, so we have dialysis. And what happens is the blood of the patient is removed for cleaning. It passes through a pump, an anticoagulant's added, which stops it from clotting, and then it enters the dialysis machine. In the dialysis machine, the fluid that's there has got the correct concentration of urea, sugar, ions, amino acids that should be in that person's blood. Anything that is in excess, so urea in particular and salts, move from the person's blood into the dialysis fluid. This fluid is then removed and new fluid is pumped in to maintain the concentration gradient. So dialysis fluid contains the correct concentrations of salts, sugars, amino acids that would be found in a person's blood, but no urea. This means that the urea in the person's blood is in a higher concentration, so it moves into the dialysis fluid. Once it's been filtered, it passes through an air detector, which removes any air bubbles, and it's pumped back into the patient. You could be asked advantages, disadvantages of dialysis and kidney transplant. Main disadvantage of dialysis, it's very time consuming. You need to have a strict diet because you can't eat salt unless you are hooked up to this machine. The main positives of kidney transplant, it is basically a cure because the kidneys will work correctly. It is less time consuming. Main negative, you would need to take anti-rejection drugs. So you'll need to take rejection drugs or immunosuppressants, if we give them their real term. Obviously, the next part of this, you need to know what deamination is, which I know my set one really struggled with. I'll go through it quickly. So what you've got protein in your diet. This protein can be broken down into amino acids. Amino acids are used by your body to make new proteins. However, if you eat too many amino acids, they are deaminated in the liver, which makes this compound here, which is ammonia. Ammonia is very toxic, so it's straight away converted into urea, which you get rid of in your urine. This would be a typical exam question on it. Amino acids cannot be stored in the body. Describe as fully as you can what happens to the excess amino acids. You might want to use the following words. If it says you might want to use them, make sure you use them. So, protein is taken in the diet. It's converted to amino acids by protease enzymes. If there are too many amino acids, these amino acids are deaminated in the liver, which produces urea. The urea is filtered from the blood in the kidneys, and then the urine is stored in the bladder. That is as easy as you would need to make it. Okay, so we're going to have to speed up a little bit. I'll get on very quickly to this next part. As well, in this section, you could be asked about plant hormones, so I'll try to save a bit of time here. So your first plant hormone is auxin. Auxin It functions in plant tropisms, as someone asked me earlier. I haven't got time to go through the auxin pathway, so how it works. If you are stuck, look in your revision guide. So auxin, depending on its distribution, will cause unequal growth rates in roots and shoots. Auxin can be used as a weed killer. That's the main way that auxin can be used. Next one, you've got gibberellins. Gibberellins are important in initiating seed germination. So the uses of these can be used to promote flowering. And the last one that we have got ethene. Ethene is a gas, and what it does, it controls the cell division and ripening of fruit. So you can use ethene to ripen fruits before you put them into the supermarket. I did say I wasn't going to draw it, but I am. If you've got a plant, it will naturally grow upwards towards the sun. If the sun is coming from one side, what happens is the auxin accumulates on the shaded side. This is because auxin is destroyed by sunlight. So if the auxin accumulates on the shaded side, what it does, it makes the cells elongate faster on the shaded side, so the plant will bend towards the light. Okay, so they're your main plant hormones. This is called phototropism. So it's a positive phototropism. You've also got gravi or geotropism. This is going away from gravity, so it will be a negative geotropism. Okay, and that's it for spec 5. Next little bit that we'll touch on is spec 6, very, very quickly. So the main part, we will have a drink. The spec 6 is about your DNA. So like Miss Ella said, DNA is a polymer. It's 2 strands twisted together in a double helix. For separates, you need to know the bases that are in DNA. You don't need to know the full names, you just need to know them as A, T, G, and C. These bond together in a specific way. So A will always bond to T, G will always bond to C, and that is the base pairing. You could be asked in an exam, like you were on your mock, to identify what is in a nucleotide. So DNA is a polymer of nucleotides. So you have got a sugar group, you've got a phosphate group, and you've got a base. Okay, so sugar, phosphate, and base. I like to remember it as we've got a house, excuse my bad drawing, it's got a satellite dish, and it's got a garage. So if you're ever asked to draw it, that's how I remember it. So your house is your sugar, your satellite is your phosphate group, and your garage is your base. That sounds really bad. So that's how I remember your DNA nucleotide structure. Okay, so we'll just move on very quickly. We'll have a look at cloning, which is, oh no, we're not. We're going to do protein synthesis. I'm jumping ahead of myself. A lot of people have asked for me to go through protein synthesis, so I will. You need to know the stages of transcription and translation. So if we have a look at it, this is all in the nucleus. What happens is your DNA molecule will unwind and unzip, so only one side of the ladder is shown. This is all in the nucleus. Excuse me. The DNA is too valuable to move from the nucleus. That looks like nucleus, never mind. What happens is, from the nucleus, what happens is, it's called transcription. You get a strand of mRNA forming, and this comes from the nucleus. So you've got an mRNA strand. Once this mRNA strand has formed, we say that it's transcribed, because this stage is transcription. And instead of your actual DNA molecule moving out the nucleus, your mRNA moves out the nucleus, and off it pops to find one of these, a ribosome. You know from Spec 1 that ribosomes are where protein synthesis takes place. And what happens is your mRNA strand moves through the ribosome, and as you know, DNA is read in triplets. So it's a triplet code. So the code on that triplet will tell something called tRNA, which is transfer RNA, to come along and bring an amino acid. That triplet will then move through, and the next triplet here will tell a different amino acid to come. Then the next triplet will tell a different amino acid to come. And this builds up a protein chain. And this stage is called translation. Once your protein molecule has formed, it then folds in on itself to give a very specific shape. And this is important because if for any reason there is a mutation in the DNA or could be in the mRNA, it will mean that the shape may differ. If you've got a different shaped protein, then it will have a different function. What's it, Miss Ellers? What spec is this? So this is spec 6. Spec 6.
Thank you.
Okay. So mutations mean that you might get a different shaped protein, which might not work. Not all mutations are negative. A lot of them don't even have any effect. In your DNA, you've got coding and non-coding parts. So if there's a mutation in a non-coding part of DNA, it might just affect how the gene is turned on and off. Okay? So not every single not every single mutation has a negative effect. Right, so we are still on spec 6. The next little bit is about cloning. So I'm just going to go through adult cell cloning. The other types of cloning are obviously taking cuttings, which is just like, planting a plant from a cutting. And I'll go through the other type as well a little bit later. So this one is called adult cell cloning. And what happens is in this, and it's how Dolly the sheep was formed. We've got 2 sheep, and I want to make 2 sheep. I want to make a clone of the sheep on this side, so the sheep on the right. What I need an egg cell from this sheep, and I'm going to remove and destroy the nucleus. From my other sheep, the one that I actually want to clone, I'm going to get a body cell. In Dolly's case, it came from an udder. I'm going to remove the nucleus, but keep it. So remove the nucleus. I'm going to fuse the nucleus with the empty egg. So the empty egg has now got the nucleus in from this sheep. We give it a little, can't draw it properly, little electric shock. You let that one cell divide into an embryo. Then you implant it in the host sheep's nucleus. What you will end up with, you'll end up with a clone of this sheep because the genetic material comes from the nucleus of the body cell. There's another part to this where you can carry out embryo transplants. So with this embryo, before you put it in anywhere, you can do an embryo transplant. And in this case, what happens is this little embryo, before you put it in anywhere, you just simply split it up a few times. What you'd end up with there is 2 genetically identical sheep rather than just one new one being formed, and that is embryo transplants. Okay, right, we are seriously going to run out of time, guys, so we need to make a decision. Do you want me to go through the theory of evolution, or do you want me to move on to the spec 7 separate content, which is decay, pyramids of biomass? So someone give me a hint. What do you want to do? Theory of evolution or decay? Do we want to do both?
We've got spec 7s, we've got a mixture of both here.
Right.
We've got quite a lot for decay.
Yeah.
It's more leaning more towards decay. We've got a good balance of both.
Okay. We'll have 2 minutes on evolution speciation, then we will split over to decay, which is really, really easy. You need to know, in evolution, you need to know about Darwin. You need to be aware of the journey that he went on and his key findings. So Darwin found that there was always variation in a population. He said that the best suited would survive and that a characteristic would be passed on. That's what Darwin said. He published his work in The Origin of Species. You need to know the date of that. That was in 1859. Not a lot of people believed Darwin at the time because he was a bit revolutionary. Plus, the main reasons, his theory challenged the idea that God created, all life. Also, there was keyword insufficient evidence. Don't write no evidence because we did have some, but there was insufficient evidence. We also did not understand the mechanism of inheritance. So we didn't know about genes, DNA, things like that. All right. Helping with this, we also need to know a little bit about a guy called Mendel. Mendel was a monk and he did a lot of investigations into the inheritance of factors, such as the colour in pea plants. So Mendel, he investigated peas, and he found out that the specific patterns, and he thought that these specific patterns or ratios must be passed down in the form of information, and he called this information units of information. So it was only in the mid-19th century that Mendel made his main discoveries. In the late 19th century, we were able to observe chromosomes. Put Mendel there. The next part in this, in the mid-20th century, we found out that units and chromosomes behaved in similar ways. And we led to genes, or it led to genes being discovered. And then the next part, again, in the mid-20th, we discovered the structure of DNA. So you could be asked about how the gene theory has developed. So this is a pathway. So in the mid-19th century, we had Mendel. Late 19th century, we discovered chromosomes. Mid-20th, we realised that the units and chromosomes were actually genes or contained genes. And then in the mid-20th century, we discovered the structure of DNA. Okay. Next little bit. As well, in step 1, you could be asked about speciation. And what I'm going to do, I'm going to draw as a little diagram. So originally, on an island, I'll just draw dots because I can't draw, there was one population. Due to a catastrophic event, the island was broken in half. So this population became geographically isolated. And in each population, there was, as Darwin said, variation. So there'd be variation there and variation there. This is variation in alleles or in genes. Over time, we've got our little island. We'll imagine one side for some reason got really hot, one side got really cold. So the ones on the hot side, they got smaller because if you're smaller, you've got a smaller surface area to volume ratio. The ones on the other side got bigger. So they have a larger surface area to volume ratio. These ones have got a smaller surface area to volume ratio. So they become suited to their environments. Sorry, excuse me. Hay fever's killing me. Right, so they become suited to their environments. Over millions and millions of years, what you can actually end up with is the ones on this island becoming so different to the ones on this island that if they ever met, they would not be able to successfully interbreed. So they would have, move it around, become different species. That is the process of speciation. Okay. Just need to be aware, as well as Darwin, there was also Alfred Russel Wallace. And he proposed a very similar theory to Darwin, and he published it around the same time. So he looked at, in particular, warning colorations on certain animals. Okay, so that's it that you need to know for spec 7. So we'll move on and we will look at this spec 6. Sorry, we'll look at spec 7 and I'll go through the main parts. So the one that everyone's been asking for, we've got decay.
Just a quick question, Mr. Nessie.
Yep.
Do we need to know about Lamarck?
Yes. You do need to know about Lamarck. If you want me to just flip that. Lamarck is the easy thing though, so I shouldn't really be asking about this, I don't think, at this point in time. But anyway, Lamarck. What Lamarck said was that over an individual's lifetime, they become better adapted. So we start off with a small giraffe, and then over its own lifetime, it developed a characteristic. So, for example, over its life, it got a long neck. This giraffe would then pass on its long neck to its offspring. We know that this is not correct because it is not over something's lifetime that it develops a characteristic and passes it on. These developments are due to mutations when over a long period of time due to natural selection the characteristics are passed on. So it isn't the fact that you become really muscly in your life and you pass it on. It's over a really long space of time due to natural selection that you would pass on any characteristics. Marx's theory was called the law of acquired characteristics. That was his theory, and we know it is wrong. We know that the right one is natural selection, which is what Darwin and really Wallace put forward. Okay. So, on to spec 7. You should be able to explain what causes decay and the factors that affect it. So if we have a look, what causes decay? Bacteria. And it's the respiration of these bacteria that will release CO2 into the atmosphere, and it's the action of digestion of the bacteria that releases biological materials back in. So you need to know what affects decay. So temperature, water, and oxygen availability will affect decay. If you increase the temperature, it gives the more optimum temperature for enzymes. If you increase water availability, it means that the bacteria can feed effectively. If you increase oxygen, it obviously means there's more oxygen for respiration. Okay? So you could be asked in an exam about a compost bin. Ideal compost bins have holes in it. You would have to say why. So it needs holes in it to allow oxygen in for respiration, but also to maintain the temperature. So it needs to be able to radiate or lose heat. Okay, that's a part of decay. So you could be asked how to design a compost bin to make sure that the rates of decay are optimized. Another question that you're all worrying about, and it's that one about how the bacteria actually decay material. So if we imagine we've got our bacteria from a loop of DNA, the bacteria will secrete enzymes. And from paper 1, you need to specifically say these enzymes. So protease, lipase, amylase. And then you need to talk about those in turn. So the protease breaks down protein into amino acids. These obviously contain nitrogen, so that would release nitrate into the soil. If we look at lipase, it will break down fat into 3 fatty acids and glycerol. I would say that the fatty acids would decrease the pH of the soil. Amylase will obviously break down starch into glucose. This glucose could then be used in respiration. Okay, so that's the main part of decay. There is a required practical for this. So the RP for decay, we looked at the decay of milk when we added lipase, and we measured the pH. If fat is digested, it makes 3 fatty acids and glycerol. Look carefully at what we made there. So we've got 3 fatty acids. If we are making acid, we are going to decrease the pH. So the quicker the fall in pH, the quicker the rate, the rate of decay. And that's the main things that you could be asked about decay. I've also got a biogas generator. Sure, if it's all right. So we need to know how a biogas generator is produced or it is used. Ignore this one for now, that's for later. I don't know why I stick them around that way. So in a biogas generator, what we have is manure This is then put into a fermentation vat. When you are making biogas, it is very different from normal decay because you need it to be anaerobic, so there is no oxygen. If any oxygen gets in there, it's going to switch to aerobic respiration. That is not what you want or need to make methane. The whole point of this is to make methane gas. This methane can then be used for cooking, heating, things like that. So in a biogas generator, you've got a cow. It does a poo. You put it into your fermentation vat, put the lid on so it's anaerobic. This will then, or the bacteria, will then ferment the material and it will release methane. So it's a closed system. So even when you are burning that biogas, eventually the carbon dioxide is used by the plants to make carbon compounds and the cow will eat it, which means that it is not contributing as much to the carbon or to the excess of carbon dioxide in the atmosphere. Okay? So that is it about decay. I'm not going to go over trophic levels, pyramids of biomass, transfer of biomass, because that is easy stuff. Just be aware that there's not usually more than 4 trophic levels because energy is lost at each stage. If we have a look very quickly, I'll come back to that picture at food production. Food production. You need to know some main ways that food can be produced. So you need to know about intensive farming. You need to know about fishing. You need to know about biotechnology. If we very quickly look at intensive farming, in intensive farming, what we tend to do is keep animals indoors and in confined spaces. If we keep animals indoors and limit their movement, this will reduce energy loss to the environment. And if we keep the animals indoors, they won't have to regulate body temperature as much. So this will again reduce energy loss. If you've got less energy being lost in movement and maintaining body temperature, more of that energy can go into building new biomass or new material that we can then use. As well, please be aware that animals in intensive farming are fed a high protein diet. This makes them grow very quickly, which again will reduce energy loss because we can get them in and out very, very quickly. Okay, last little bit. Fishing. What have humans done to fish stocks? We have decimated them, so there is much less fish in the seas. What can we do to try and limit that? We can impose fishing quotas. That is that you can only catch so much fish or a massive fish to try and limit how much is being taken out of the ocean. We can also control net size. Sneeze. All right.
Would you like another tissue, Miss Messi?
Just going to push through. Right, so impose fishing quotas, control net size. With the controlling of net size, you need the net to be big enough, or the holes to be big enough. That sounds awful. Ugh. Big enough to let out the small, young fish. If we let the young fish out, they have got time to breed. Okay, So we need to reduce the amount of fish we take out the oceans. We can do this by imposing fishery quotas and controlling net size. Last little bit, biotechnology. You should all, hopefully, once in your life, have tried this stuff, corn. Corn is a food that's produced through the use of biotechnology. And what it is, corn is a mushroom or a fungi called fusarium, and you need to know that name. What you do, you grow it in a fermentation vat, but the main difference between this is that this is aerobic. So this process needs oxygen. So you put in your fungi, your fusarium, and you also put in glucose syrup. The glucose syrup provides glucose for respiration, so for respiration, and also for building biomass. So the fusarium uses the glucose in respiration, it grows, and what you do, you bubble sterile oxygen through, so it keeps it aerobic. You also need to monitor the temperature and pH to ensure that it's at the optimum. Once the fusarium has grown in here, you harvest it, purify it, and you can turn it into corn. Okay, so this is one role of biotechnology, so using fusarium to make corn or substitute meat products. The other one, which Miss Sellers has gone through already, is genetic engineering. I just want to mention to you a few things. Golden rice. What's funny about that?
Where did that come from?
You need to know it.
Golden rice.
Golden rice. So golden rice has been infused or genetically engineered, rather than making a cup of tea. It has been genetically engineered to contain beta carotene, which can be converted into vitamin A. So in a lot of developing countries, there's a lack of food with vitamin A in it. If we can genetically engineer rice to contain vitamin A, it can save a lot of children's sight. Okay, so uses of biotechnology, you've got making corn, you've got genetic engineering. The only bit that I have not gone through with biology only stuff is the trophic level stuff and pyramids of biomass. I think year 11 top set, you can quite effectively cope with drawing a pyramid of biomass. Just remember, the producer is on the bottom. You're not going to get more than 4 trophic levels because of the energy loss between each stage. Okay, any questions?
Just give me a second. I think we've had. Nothing through the minute. There's been quite a bit of scroll by, so you can see anything.
So, any questions?
Thank you for giving it your Sunday evening and staying on longer than you said. I'm Dylan Sargent, so thank you.
No worries, Dylan. Always a pleasure. You're going to get me a grade 9, so I do not mind at all.
What does vitamin A do?
Vitamin A, oh gosh, you asked me pretty much. So I learned it in my degree. You don't need to know the actual functioning of vitamin A. You just need to know that it can help protect people's eyesight. So it's involved, I believe, in producing the cells in the retina.
Do we have to know the dates for Darwin?
Yes. 1859.
Is Miss Neresi going to book a doctor's appointment?
Well, funny story. I'm pretty sure I told you about. Is that Megan?
No, it's Lauren.
Lauren, I've been trying. I promise I'll do it on Monday. I promise.
What is a recessive allele, miss?
A recessive allele, the definition you need to remember, to show a condition, 2 recessive alleles need to be present.
Do we need to know about differences between rock and mold fossils?
Absolutely not.
So we've got quite a few students from different schools, and different parts of the world watching as well. Actually,
What you need to know, well done, lovely. What you need to know about fossils is just the way that they form. So you can get imprints in rocks. Parts might be replaced by minerals, and like Miss Sellers said, sometimes hard parts do not decay.
How is the carbon recycled into the growth of new leaves?
Right, so thinking about the carbon cycle, I've lost my picture. Oh, it's here. I'll just get it. Into the growth of new leaves. So if we look in the carbon cycle, any carbon that is released in respiration is then taken back in by the plants to use in photosynthesis. The carbon dioxide forms with water glucose, and this glucose is then converted into protein or cellulose. Protein is used for building, cellulose is used to form cell walls. Okay?
Just a quick question about required practicals. Is there any for separates only, or is it just those two for sampling and reaction times?
Pretty sure I went through it.
No, some people are tuning a little bit later, I think.
All right, I'll get my clothes back in. Yes, separates you need to know decay. The decay required practical. Is that it? Yeah, the decay required practical.
Do we need to know what the different female hormones...
Oh, sorry, wait. Decay and plant hormones. I always forget about that one. You've got your plant hormones one.
Do we need to know about what the different female hormones inhibit or stimulate?
Yes, you do.
Is it worth slipping a 20 in my paper to get some extra marks? Probably not.
Who was that from?
I'm not. I've missed that one. That's just scrolled up. How blurred vision is corrected?
If you're talking about myopia and hyperopia, yes, you need to know. So myopia, you're going to use a concave lens. Hyperopia, it is a convex lens.
How many practicals are there in total for paper 2? Is it just those 3?
So for Trilogy, it's 2. Separates is 4.
Do we need to be able to draw a convex and a concave lens?
You could be asked to draw ray diagrams. It's unlikely you'll be asked to draw the lenses. When you're drawing ray diagrams, remember pencil and ruler.
What are the main things to learn for foundation?
If you're doing foundation separates, that's not something that we offer for Horizon. So you would need to check with your teachers at school by tomorrow morning.
Any chance to get your last couple of questions in, ladies and gentlemen, before we turn off before Love Island after multiple requests, and we will upload for you. Is glucagon involved when glucose levels are too low?
Yes, it is. Glucagon is released when all the glucose has gone, so glucose levels are low.
Can you please show us how to do Punnett squares? We've already looked at Punnett squares, so you will need to watch it back once we upload it afterwards. What would happen in the body of a person with hyperthyroidism?
Right, you need to be aware that the thyroid is involved in, metabolism and respiration. So if it's hyperthyroidism, there'll be too much thyroxine made. If there's too much thyroxine, respiration rates will be extremely high, so the person will be likely to have a very low body weight. But that'd be a grade 9 question.
Do we need to know about the Human Genome Project?
You need to be aware of it, so you need to know that the human genome has been sequenced. And like Miss Sellers said, you need to know the impacts of that, so it helps us when looking at inherited disorders, migration patterns, things like that.
Is this still Trilogy? No, this is separate science, but there's some questions for Trilogy chucked in at the moment. Amelia, do we need to know that DNA is made up of nucleotides?
Yes, you do. You need to know the structure of a nucleotide, so you need to know the little house, the satellite dish, and the garage.
Do you think grade boundaries will be low? We've no idea. The grade boundaries are decided after all the exams have been sat, so we have no idea. What do you think will be in the paper tomorrow? No idea. This is the first year this paper has been sat with it externally, so it could be anything on there. We're not too sure, and we're not going to make any predictions because it is literally just guesswork. Yeah. What are the key things that are very important for this exam? Definitions are probably a key one, and everything. But definitions are
definitions in biology. You're looking at maybe 20 marks on a paper that are linked to definitions.
Okay. Any more questions? We're turning offline at 45. So you've got last 2 minutes. Could you please explain glucose and glycogen, etc., please?
We've already gone through what Miss Sellers did, so when we upload the video, you'll have to rewatch it back. We are going to have to go offline now because me and Miss Stellas need to get home, see our families, you know, that kind of thing. If you are desperate, email me, set1@Horizon, you know, my email address, and I'll try and get back to you tonight. Obviously, I will be going to bed soon.
Just so you know as well, I've put on a couple of times, but we will be uploading one for chemistry and for physics. So if you if you subscribe to our channel, click on the notifications, turn them on, you will get a notification through whenever we post a new video. Also, if you follow our Twitter page, which is at HCC_depth of sci, we also post links onto our Twitter page as well. So good luck, Year 11, whether you're from Horizon or any other school in the UK or schools in other parts of the world. We wish you all the best for your exam. Thank you very much. Bye.
Bye-bye.